Thank you very much for having me. I'm really excited to talk a little bit about the work that we are doing in an international consortia who basically recruit colorectal cancer patients at the time of diagnosis. And I have to apologize for the short-term change in my title, but I thought it would be more interesting if I talk about the metabolomics and the application in colorectal cancer research, and then talk about the different, the different outcomes we're working on. So what we do in our studies so far, we have used the P180 targeted metabolomics kit. And what we're doing, we're looking at biomarkers for cancer risk and cancer prognosis. And as you may all agree here, really the most important thing is to understand cancer metabolism. But metabolites also find application in drug development or in monitoring treatment responses, or when we talk about nutritional interventions. So again, we used the targeted metabolomics kit. It was measured at the International Agency for Research on Cancer in the lab from Augustin Scalbert. And I think this work is now being continued by Pekka Keskirakonen. And after cleaning and harmonizing the data, we identified 130 metabolites that we could use for analyses. These include amino acids, biogenic amines, which we measured via LC-MS, and we used internal standards to the lab, but we also provided samples from our own cohort as a specific quality control across all of the batches that were analyzed. Other ones include the acylcarnitines, hexoses, phosphatidylcholines, and sphingomyelins. And particularly the last two, I have the feeling, show up in a lot of studies, which is showing their importance, particularly in the area of cancer research. Yeah, so we have the data will be pre-processed. We have a specific approach for handling missingness, and we usually either do a log transformation or a C transformation to bring the batches of the different cohorts together. So this is the ColoCare study. She's the basic for all of the study I'm doing. It's a prospective cohort. We have almost recruited 4,000 patients, have NIH funding. And we not only collect like plasma, serum, and questionnaires, but we also have access to tumor and normal tissue, stool samples, body composition data. So it's a really rich resource. And since it's U1 funded, everyone is invited to have a look and see if there are data you can work with for your own research project. So we're really happy to share. This is the project that I will be talking about today. It's the MetaboCCC European Consortium. And as you can see here, it's really international. We have the Netherlands, the US, we have Norway, we have Austria, and a study site in Heidelberg. So as you can see, there are a lot of different international locations, but this comes with specific challenges. For example, if we think about the diet and looking at the association of diet with metabolomics, it's getting a bit tricky, but I think like having these pieces from all different parts of the world will make it so a biomarker that survives the analyses is very robust because you see it independent of all of the differences that we have across the sites. Here's our team. We get Transcan funded and to measure the metabolites at the International Agency for Research in Cancer. And this is one of our team meetings that we had in Europe. So just a brief explanation. So MetaboCCC has 4 European cohorts, as I mentioned earlier. So Germany, Austria, and 2 sites from the Netherlands. We have a pretty impressive number of patients, with participants. So it's over 2,100. 800 are about colorectal cancer. Then we have colorectal adenoma patients. And what I think is very unique to this project is that we have 753 cancer-free controls. We did targeted and untargeted metabolomics for this data set, but today I'm going to talk about the targeted data. And the specific research project that I want to introduce to you today is one where we had 300 ColoCare patients and 150 cancer-free controls from the Heidelberg cohort. So the project I want to talk with you is visual adiposity and how it's associated with the aggressiveness of colorectal cancer tumors. And I think that's really an emergent field. There are a lot of data out there, but still we don't fully understand what is the underlying mechanism of obesity or visceral adiposity who's really leading to this increase in cancer risk, but also worse clinical outcomes for our patients. So what we know is that visceral adiposity is an established risk factor for colorectal cancer, particularly as adipose tissue is more than just an energy storage organ. So there are a lot of different other roles. For example, it secretes cytokines, adipokines, and hormones, which is of particularly importance for postmenopausal women. If they have like high adipose tissue, they have a higher risk of developing like a chronic inflammation. And what we also know that adipose tissue is really heterogeneous. So we have visceral adipose tissue and we have subcutaneous adipose tissue. So if you look at these CT scans, so this is basically where we're using a color coding to identify the area of visceral adipose tissue and the one for subcutaneous adipose tissue. At this point, we don't do any work on brown adipose tissue, but I think that would be a really exciting thing to look at for the future. One of the other problems that we have is that BMI is the most commonly used proxy for body composition in almost all cohorts across the world because it's really easy to determine the BMI.
Ich sehe Ersilio Esito German nicht in deinen Kontakten.
Wem
möchtest du eine Nachricht schicken?
Ich will niemanden, Moment. Okay.
This happens sometimes.
Entschuldigung. Okay, so the main problem that we have is that with the BMI, you can measure obesity, but you cannot quantify adiposity, right? So BMI is basically not a very good measure to address the differences in our body types. Again, the visceral adipose tissue, subcutaneous adipose tissue, and most important, muscle tissue, sorry. So the aim of this study was really to use this targeted data that we have on our plasma metabolites and really look at multiple dimensions of body fatness in early and late stage colorectal cancer using patients enrolled in the ColoCare study. Briefly, the method, and you may already have realized that my presentation is really like molecular epi oriented. So what we did is we identified 288 colorectal cancer patients diagnosed with stage 1 to 4 tumors. What's really important to say about this study, we had very well established SOPs on how to collect, process and froze the samples that have been collected within 4 hours after collection. We used the P80 kit, amino acids, biogenic amines, glycerophospholipids, and so forth. And what I think is really important for the work that we are doing here, particularly if at some point we want to pool data from different cohorts together, is to have quality controls that we can use. And there are biocrates internal ones. Then we have lab QCs from the National Agency for Research on Cancer. And then actually we have our own quality control samples that we try to measure with every metabolomics project that we are doing to have the opportunity to then again get those data together. Intra and inter CVs were excellent. They were all below 20%. And then we basically used clinically available CT scans to quantify the adipose tissue and skeletal muscle mass areas. We did this on 2 levels. And again, we were looking at visceral and subcutaneous adipose tissue. We also did partial correlations and regression analyses. It was particularly exciting to develop the adjustment model for this. So for now, we are going with age, sex, batch, and tumor stage. We will not adjust for study site here because the assessment by batch is basically a proxy for which study sites the samples are coming from. Then we did the stratified analyses to really better understand the metabolomic profiles for early and late stage tumors. And of course, we have corrected for multiple testing using FDR. So this is the study population. Each line that each underlined item means that there are statistically significant differences between non-metastatic cancer patients and metastatic cancer patients. So non-metastatic is stage 1 to stage 3, and metastatic is actually stage 4. So we see significant differences by age. We also see significant differences by BMI, which is kind of expected. We didn't see any differences by tumor site, which is kind of a good thing to know, but yeah. And then we have adjuvant treatment, which was significantly different. And I'm not sure why this slide stops here, but we also didn't see any significant differences for neoadjuvant treatment. This is the description of the fat areas that were quantified using these CT scans. And we have, like, everything again that's underlined here was statistically significant. So we see that for the visible fatty adipose tissue on level L3, L4, L4, L5. So the L3 and L4 does not matter at this point. We just try to do comprehensive analyses. This is why we looked at 2 different level types. And then we also saw difference for VFA at the L4, L5 level, and also for the subcutaneous fatty area. So we see there's already something happening between patients that are non-metastatic and patients who are metastatic. We identified 127 metabolites from 5 different compound classes. We looked at overall obesity and subcutaneous adipose tissue, and they were not associated with metabolites overall or an analysis by stage after FDR adjustment. We also identified 7 glycerophospholipids, and I think they're coming up more and more in the field as really important biomarkers that were inversely associated with visceral adipose tissue. 1 second. So this slide is needs a little bit of guidance. So basically what we did here, we looked at the 2 patient populations separately. So what you can see here to your right hand is the early stage tumors. And what you can see on your right hand is the late stage tumors. And the more intense the color, the more intense the correlation. And here we have the visceral adipose tissue on 2 levels and the subcutaneous adipose tissue on 2 levels. What you see is that we have really strong correlation for the visceral adipose tissue. But if you look at the subcutaneous adipose tissue, there's basically no significant association for correlations of the metabolites with the subcutaneous adipose tissue. The interesting part here is then when we look at the stage 4 tumors, so you can see here we have this strong correlation, and it continues to be strong even for metastatic tumor. But the really interesting finding that I'm getting out of this is that for early stage patients, there's no association of subcutaneous adipose tissue with metabolites. But this entirely changes when you look here at the late stage tumors. So the association is basically only relevant for the metastatic tumors. And most of those that we saw here were glycerophospholipids, lipid metabolites, and so forth. Okay, yeah, again, so glycerophospholipids really showed up. We know there are major components of cellular membranes. They're really pertinent to cancer cells that undergo transformation, progression, as well as metastasis. And also, like, highly proliferating cancer cells really need to synthesize fatty acids de novo. And in order for doing that, they need the glycerophospholipids for the membrane production and making sure that everything is, like, biologically healthy. But one thing that I forgot for this slide here, the hypothesis that we have here is that as soon as the patient, the tumor reaches the metastatic stage, right? The tumor is, like, at various parts of the study, then we see this correlation with the subcutaneous adipose tissue. And and we actually hypothesize that the subcutaneous adipose tissue is more important in metastasis because it's basically giving fuel to the tumor directly. And we still need to think about the biological rationale, but this is one of the ideas that we had. So in conclusion, we saw strong negative association between the visible adiposity and the glycerophospholipids. But and we also see that, let me 1 second here. And we also see that there are differences if you're looking at late stage and early stage tumors. The results that we have suggest that there's really like a metabolic shift in the glycerophospholipid metabolism, which is related to subcutaneous adipose tissue, particularly, sorry, in metastatic tumors. So the next steps are to expand the sample size, to have more statistical power, and also to perform survival analyses for this area. So these are just like a couple of examples what we can do. So we already did a study where we looked at, where we basically looked at the metabolites and then see how they were associated with patient death. So we used the targeted metabolomics. We have 680 patients, which is really a lot if you think about all of the patients that have to be recruited and samples need to be collected. And 14% of these patients deceased, and we did stratified survival analyses by tumor site. We also did a metabolite set enrichment analysis. So what we found out is systemic creatinine is associated with an increased risk of this. But this is only for the rectal cancer patients. We didn't see that in the colon patients. So creatinine is a breakdown product of creatine, which promotes metastasis, but it's also very relevant for muscle metabolism. The pathways that we identified were the trialose degradation pathway, and it was also associated with increased risk of death, with an FDR-adjusted p-value of 0.018. Okay, this one is not looking at data from blood, but this is a study where we did use urinary metabolomics. We identified branched-chain amino acids and saw how they were related to energy balance. So these branched-chain amino acids were not associated with energy balance over time. The catabolites of the BCAA metabolism were notably inversely associated with risk of death. So that is something we still have to wrap our head around. And then obviously it plays a really important role in prognosis and physical independent of muscle mass and physical activity. This is a really, really small pilot study, but we needed it to understand if we can even do cachexia research with the data that we have available. So this was a pilot study with 16 cachectic, 13 precachectic, and 23 non-cachectic patients. So we identify a set of metabolites that very well discriminated cachectic from non-cachectic cancer patients. And I think it makes a lot of sense when we see now that the glycerol phosphate shuttle metabolism and glycine, as well as the serine metabolism, are related to cachexia. And we're currently working on expanded this project to patients who are part of the large ColoCast study and the Metabo-CCC cohort consortium. And of course, we're really excited also to give access to data. If you have ideas, you can contact it. So this is one where we looked at the plasma metabolites and recurrence. So just to show you how well usable we can use this data. And there we had 440 patients diagnosed with stage 2 and 3 colorectal cancer. Yeah, identified 128 metabolites from the pre-surgery plasma collection, and 30 metabolites were nominally associated with a reduced risk of recurrence, but after controlling for multiple testing, none were statistically significant. If I would this study again, I would not use the classic cutoff of 0.05, but do it like other colleagues in the field. and use either 0.10 or even 0.20 to adjust for the multiple testing. Also the pathway topology analysis that we performed did not really reveal significant association. And what you can see here, I'm not sure if you see all of the slides, but we have reasonable statistical power, and really the phosphatidylcholine, sphingomyelin, they were all associated with a reduced risk of recurrence. So there are definitely some things that we can do to better understand the biology behind it. As you can see, none of the FTR adjusted p-values were significant. A big thank you to the colorectal study team, my mentor, Nelly Ulrich, all of the patients who provided their samples and filled out questionnaires. And if you're interested in a postdoc or research scientist position in Utah, please let me know. We're currently looking for new members for our team. Thank you.